from some short answers from previous exams. And I'm going to do this on the document camera. And this first one that we're going to go over was from 2010, so it's one of your practice exams. And it says, answer the following questions about cellular respiration based on the drawing below. So here's one where you just have to pull some of these words out of memory. What is the name of the process in step A? Shout outs. Glycolysis. Very good. Is glucose being reduced or oxidized? Oxidized. Very good. I still have to think every time when I write this, every time. And sometimes in the lecture, when you hear me pause, it's because I'm thinking, oxidation, oxidation is losing electrons. Okay, what process is taking place in location B? Citric acid cycle. What is that location called? Right, it's the mitochondrial matrix. Name the molecule that's the final electron acceptor in C. Oxygen is correct. And what molecule then does this make? It makes water. Where did the electrons come from? They came from glucose. The electrons were then donated to the electron transport chain, and that powered the hydrogen, the pumps in the electron transport chain to make a hydrogen gradient, right? How would decreasing the pH of the intermembrane space affect the rate of chemiosmosis? This one is a bit more tricky. Talk to each other for 30 seconds, and I'm going to ask you to vote. Okay. How would decreasing the pH of the intermembrane space affect the rate of chemiosmosis? Raise your hand if you say it increases the rate. Raise your hand if you think it decreases the rate. Okay, it's about 50-50, maybe a few less say decrease. Okay, so let's first talk about if you have a decrease in pH in the intermembrane space, what are you going to do to the, what is happening to the concentration of hydrogen ions? Okay, it increases. I heard some of you say decreases. It increases. So if you said decreases, you know that's where you went wrong in this problem. Okay, it increases. So when we have high levels of free hydrogen ions, right, this is acidic. Okay, and so now, how is this going to affect the rate of chemiosmosis? Remember, these are the hydrogen ions that are in the intermembrane space. Is it going to increase the rate of chemiosmosis? Raise your hands. Decrease. Okay, more saying increase than decrease, which is correct. It will increase the rate of chemiosmosis. Because what happens to those hydrogen ions in the intermembrane space? Where do they go? They flow down into the matrix through what protein complex? ATP synthase, right? So they go down through ATP synthase, and that then combines what two molecules? ADP and PI to form ATP. Okay? So if you increase the hydrogen ion concentration in that region, you made it more acidic, right? And you have more hydrogen ions to flow down, and so the rate of chemiosmosis goes up. Okay. Moving on to the next one. Okay, I'm going to read this one to you. SGLT1 moves sodium ions down its concentration gradient, carrying a glucose along with each sodium ion. Sodium ion concentration in epithelial cells is regulated by the sodium-potassium pump you learned about in class. On the diagram below, show the molecules that are moved by SGLT1 and the sodium-potassium pump, and use arrows to indicate the direction of movement. So we're going to look at that part first. Okay, so how many of you know what SGLT1 is? Yeah, most of you don't know, because I didn't talk about it. So that means you don't need to know what this is. It's actually the sodium glucose transporter, co-transporter. It works quite a bit like another co-transporter that you've learned about. What's that called? The sucrose hydrogen ion transporter, right? So, but for this question, you didn't need to know that. So people read this question in the exam, they start to freak out because they think, oh my God, I don't remember SGLT from lecture. But in fact, read carefully, and you'll see you'll be able to answer all of the first 4 questions, first 4 parts of this, with knowing nothing other than what's written here. Okay, so we know the sodium-potassium pump, right? So here, where the diagram is, this is the gut lumen. Here's your epithelial cell, and here's the extracellular fluid. And so we give you this, we tell you the SGLT1 transporter is here, in between the gut lumen and the epithelial cell, and then sodium-potassium pump is here. So let's start with the sodium-potassium pump. You know what's being pumped by this, right? So which directions are sodium ions being pumped? Out of the epithelial cell into the extracellular fluid, right? So for the sodium-potassium pump, we have sodium is moving out. How many sodiums for each? 3. Right. And then we have potassium. 2 are going in. Okay, so it says I'm supposed to be drawing arrows indicating the direction of movement. So now I get 2 points by writing this. Sodium goes out and potassium goes in. Okay, next one is SGLT. Tells me that it carries glucose, it moves sodium ions down its concentration gradient, and we're pumping sodium out here, right? So which direction do you think sodium is going to be moving? Into the cell. And what else is coming with it? Glucose, because I told you that. It said here, it's carrying a glucose. Okay, so now you've gotten the 4 points from this. And a lot of people just left this one completely blank. So I think it's because you look at it and you feel like, I just don't know where to start. So start small with the things that you know about, and then branch out to the things you feel less comfortable with. Now, the last part of this problem was a little bit harder. If SGLT is blocked, but the sodium-potassium pump works, how will this affect water flow from the gut into the epithelial cells? So go ahead and talk among yourselves in just a minute, and I'll ask you to vote. Let's go for this. If SGLT is blocked, how will this affect water flow from the gut to the epithelial cells? How many people will say it increases? Raise your hand. How many say decrease? Okay, a lot of you said decrease, and that actually is the right answer. So let's go over it. If you block SGLT, then you're going to block sodium flow down the concentration gradient, and that's going to block glucose going in, right? So now, before we block that, you imagine in equilibrium, right? There's going to be a certain rate of water flow across these. And now we're blocking this. We're blocking sodium going in and glucose going in. So the solute concentration in the epithelial cell is going down or up? It's going down. So now it is, what is it? Its tonicity with respect to the outside of the cell. It's hypotonic, right? And so it's hypotonic, so if you had water flow that was going from the gut to the epithelial cell, then you will reduce or maybe even reverse that water flow, right? Because you're going from a place of high water, relatively high water concentration to low water concentration. So it will decrease water flow from gut to epithelial cells. So this is one where you're thinking sodium-potassium pump and you're really not sure about this other transporter, and now you find yourself in an osmosis problem, right? So you have to see how all of these things are interconnected. And so don't be thinking narrowly when you're looking at these problems. Okay, and moving on to the next one. answer on it. Okay. The graph below shows the rate of G3P synthesis in plant cells over a 10-minute period in 3 different conditions. Explain what could underlie the change in rate of G3P synthesis when cells are grown in pure oxygen instead of air. The reason I'm including this one is for our wonderful TAs, because what we want you to be able to do is answer this question with one sentence and be clear and concise. Don't write a paragraph. So I'm going to want you guys to do this with the people around you on a class card, number 13, and you're going to turn this in and try and answer this question. It can be answered and you get full credit in a single sentence. And it's not even a long sentence. Okay, go ahead and finish up your cards and then pass them to the aisles and the TAs will come to pick them up. Bless you. Okay, so we know from looking at this graph that in pure oxygen, what happens to the rate of G3P synthesis? It goes down, right? And so what could account for this? Why is this happening? Yep, I heard some people say it. So what are we lacking that's in O2 that they need? Carbon dioxide, right? So in pure O2, there is no CO2 available. And where do we need the CO2? In the Calvin cycle, yeah. So, I mean, saying you need it in photosynthesis is okay, but really we're asking you to be as clear and specific as possible. So it would be no CO2 is available, so carbon can't be fixed in the Calvin cycle to make G3P. Okay, so it was important that you knew that attributed there no carbon dioxide, so now you don't have any carbon, and therefore the Calvin cycle doesn't have anything to actually make G3P. Okay, let's move on to the next one. Okay. This is a pretty straightforward one. Name the secondary structural elements indicated in the drawing of the protein shown below. So A is what? Alpha helix or alpha coils. Both of those would work. And the name of the structural element shown in B? Beta sheet or beta pleated sheet. Either of those would be acceptable answers. So like there will be answers, there will be questions like this on the exam where you're basically just answering a single, writing in a single word. That's perfectly fine. Oftentimes then that will be associated with other questions that are like at a slightly deeper level. So here's one with the S bond. What type of bond is that called? Disulfide bond shown is broken. So this bond is broken. The protein becomes non-functional. Interaction with what structural element in the cell could potentially restore function? Right, chaperonin protein complexes. And chaperonin would be a perfectly acceptable answer, but that would be acceptable as well. Okay. Okay, so that's an example of something that's very straightforward. And if you do your short answer second and you didn't get to that one, it might be the last question. That's not a good one to miss, and so go ahead and make sure that you skim through them at least in the beginning. Okay, and here is a short answer one that has a little bit more figuring that you need to do for it. This one, if you examine an epithelial cell with 1 sodium-potassium pump in its membrane and the starting conditions of 10 sodium ions and 10 potassium ions inside the cell and 10 sodium and potassium ions outside the cell or in the extracellular fluid. If you let the pump run for 2 cycles, how many sodium ions will you have inside the cell and how many potassium ions. So take a minute or two to figure this out, and then we'll go over the strategy. Okay. Number of sodium ions that will be inside the cell. 4 is correct. Number of potassium ions that will be inside the cell. 14 is correct. Okay, do you want me to go over a strategy for doing this? Raise your hand if yes. Okay, there is a reasonable number. Okay, and this is how I do it because, again, I say I can't do it in my head. So I just start out by making a chart that would say, okay, what do I know? I get sodium in, sodium out, potassium in, potassium out. And my starting conditions. And I'm told there's 10 ions in each of these. 10 in, sodium in, 10 sodium out, etc. So after one cycle, how many sodiums in do I have? 7. And how many sodiums out? 13. How many potassiums in? 12. And potassiums out? Okay. After 2 cycles. So we're taking 3 out each time. So what do we have here now? 4. Okay. Adding 3 each time, 16. Adding 2 each time, 14. Taking 2 out each time, 6. Okay, so by doing a chart like this, you could answer any permutation of the questions I would ask. And I don't always ask what's inside the cell. I do what's outside the cell. What other property could I ask about that is generated by the sodium potassium pump? Voltage, right? What is the voltage across the membrane when we're in our starting conditions? 0, right? There's no difference between in and out. After one cycle, is the inside more negative or more positive than the outside? More negative. And you can even calculate how much more negative, right? So then you can add up how many positive ions are in. 19. How many negative positive ions are out? 21. Okay? And then what happens after 2 cycles? It's even more negative, right? Because we have 18 positives in and 22 out. Okay? So anytime you're asked a question like this, you'll often have multiple choice questions on this, and they'll ask a variety of things about sodium, potassium, voltage. Those are all the things that you need to be able to answer. Okay. Now let's do... Okay, now we're going to do some multiple choice questions. And the strategy, again, I recommend strategies, but you have to try them, or you can try them. And if they don't work for you, that's okay. I'm just giving you strategies that work for me and that I've heard students say work for them. So strategy for approaching multiple choice problems that ask you to predict something based on data or a change to a normal system. You need to read the stem of the question very carefully. Figure out what you know from the data given or how the normal system functions. So list your known properties, draw diagrams. You can draw all over that sheet. And then look at the answers and find one that matches your answer. Okay, so we're going to go and do one. I'm just going to do it on here now. Just top sheet. Okay, here's the first one. Okay, so this is a multiple choice one, but I'm not giving you the choices yet. Umbilical cord blood stem cells are placed in solutions with different concentrations of solute, and the results are shown in the figure below. Given these data, in what solute concentrations is the cell hypotonic to the solution? So talk about it for about a minute, and then we'll go over it. I look to see over here on this graph, I'm going to put things into groups. So here we're looking at the percent change in stem cell volume. I see we have one group where the cells get bigger. So water must be doing what? Going in or out of the cells? In. We have one group where there's no change, and one group where the cells are getting smaller. Okay? So I'm going to draw 3 beakers. This first beaker has these 2 concentrations, 100 and 120. And what I know, if I put my stem cell in here, I know that the cell volume is getting bigger. And so I know, just like you said, the water is flowing in. And so, is the stem cell the stem cell hyper or hypotonic to the solution? Hyper. Okay. So now in this one, 140, we have our stem cell. It's not changing in size, so isotonic, right? So stem cell is isotonic to 140. Okay, and in this one we have 160, 180, 200. Our stem cell gets smaller. So which direction does water flow? Good, cell volume decrease, so water flows out. Oops, out. So the stem cell is hypotonic to the solution. Okay. Based on that, now you should be able to, one of the answers is going to ask about one of these properties. Okay, so let's go to the forward here. Laptop. Okay, and go forward. Yep. Okay, so now you're going to get to vote. All right, click in if you have not done so. Outstanding. So the vast majority you got this correct, and you can see this was a question that I think only 40% of the people got correct on the year this exam was given. And so it seems sort of straightforward now, but you will be getting osmosis problems that are using the same principles, that each time they sort of appear, they will appear in a different format, but they're using the same basic principles, so apply those basic principles. So, let's see. Oh, okay, so that's the right answer, and, okay, so here's another strategy when you're trying to figure out how to, how do I know enough, if I know enough about a topic, find other problems on the same topic. From old exams, examples in class, examples from the book. So any one where you're going to be able to actually get a bunch of different strategies to be able to look at the same problems. Okay, so I'm going to give you another set of problems. Yeah, please. And actually, this one is another one on osmolarity. And TA in Bio-93 studies limb regeneration in the axolotl, a freshwater salamander from Mexico that can regrow lost or injured body parts. Human cells are cultured in DMEM, a solution of salts, sugars, and nutrients. However, axolotl cells must be cultured in DMEM that has been diluted to 60% strength. A possible reason for this is that axolotl cells, so go ahead and just think about that, talk to each other for a minute, and then we'll go over it. Okay, let's go ahead and do this. My strategy here is just going to draw, again, sort of two beakers. In one, we have DMEM. This is, the cells are being cultured. They can be in a dish, they can be in a beaker. I just want to illustrate a solution, which is DMEM. And we have our human cells in this. And so when the human cells are sitting in DMEM, what's the tonicity difference between them? They're isotonic, right? So water flow is basically in and out. So so the human cells are isotonic with DMEM. Okay, and we have over here 60% DMEM. So what we do is put a bunch of water in, so we dilute down the DMEM, so it's a lower solute concentration. And the axolotl cells are perfectly happy in here. And so are they isotonic to the 60% DMEM? Yeah, right? So there's water flow that just goes back and forth. So axolotl cells are isotonic to 60% DMEM. Okay, so now by inference then we know, and we also know that 60% DMEM, because it has more water, is hypotonic to DMEM, right? Okay, so therefore, we know that axolotl cells are hypotonic to DMEM, right? Because they're happy in the solution. So axolotl cells are hypotonic what is the tonicity of the inside of an axolotl cell compared to the inside of a human cell? It's hypotonic as well, right? So they're not only to DMEM, but to human cells, because human cells are perfectly happy sitting in DMEM, so you know their tonicity is the same as DMEM's tonicity. Okay, so now we're going to go to the laptop. And here are your choices. Okay, click it if you haven't. Okay, 75% of you got the answer correct, right? That was one of the things that we figured out in our march through that problem. So it will decrease in volume at 100% DMEM because we figured out that it was hypotonic for that reason. Okay. So the thing is, I pick out the big themes, and the big theme that we were looking at both of these was, I had fluidity on last slide, clearly that's not right, these were osmolarity questions. So find other osmolarity questions. The marathon runner example from the class, where they lost salt from the blood through sweating, and they rehydrated with water, and so the blood was hypotonic to the tissue, water flowed from the blood into the cells, so the cells swelled, tissue swelled, constructed airways in the lungs. All of those are osmosis questions. Okay, so try and figure out all of the questions that we've been talking about and how they're related to the different major topics like fluidity, osmosis, transport, etc. And that will help you understand the sort of what we call topic space that you need to understand to be able to do well on these exams. And mostly, well on these exams as a byproduct, what we really want you to do is understand this material, because this is the basis for the medical or healthcare career that many of you guys are interested in. Okay, next problem. Two types of cancer cells, A and B, found in the pancreas differ in the total concentration of phospholipids in their membranes that are unsaturated. Which of the following statements is most likely to be true? And again, I haven't given you any of the statements. So look at this, and then we'll go through what we know from this graph. Okay, let's go through what we know about this. When I do a problem like this, almost always you're going to be asked to compare, A is more than B, B is less than A, etc. What I do when I tackle problems like this is I choose one of them. So I'm going to start with B. It's big, it looks like percent unsaturated phospholipids, and I know that it has a lot. So B, what do I know about B? We know it has more unsaturated phospholipids. What else do we know? What do we know about its carbon double bonds? Does it have more or less than A? Okay, more C double bonds. Is it going to be more or less fluid than A? More, very good. More fluid than A. So is it going to, a cell that has this membrane composition, is it going to move faster or slower? Faster. How about proteins in the membrane that are transporting ions across the membrane? Are they going to move faster or slower? Faster. Very good. Okay, so that's what you know from looking at this. Now look at your answer choices. Ooh, keep going. Yeah. Okay, click it if you haven't. Okay, outstanding. That is the correct answer. And again, this one was one that was really hard. I think again only about 45% of the students got it right when it was on the exam the year it was, which is in 2009. Okay, so try this one on your own. This is the last one we're going to do. Okay, click it if you haven't. Okay, so really this is it's almost exactly the same question as what we just asked and that we went through. But because we went through it as a class and we went through each step, you guys got it right, whereas this one, the majority of you did not get it right. In fact, this is sort of what the distribution was like when we gave this on an exam. Okay, so lower concentration of phospholipids with double bonds in the tail. We're going to have a higher concentration, right? So cell 1 went faster, right? Because the distance is greater. Okay, higher concentration of motor proteins. They're not actually really involved in this. Lower concentration of cholesterol. Could be, right? You have a high concentration of cholesterol at 37 degrees, and it makes things less fluid. Higher concentration of unsaturated carbohydrates. Phospholipids. Carbohydrates